So human microbiome technically means the study on the microbial population community on humans. So why is this important? Because now we know that bacterial cells outnumber human cells by 10 to 1. However, this is only in abundance and not, of course, not in mass, because human cells are a lot larger than bacterial cells. Because of the abundance of bacterial cells on our body, and because that now we understand that there are some interactions between bacterial and our human body, human biology can no longer concern itself only with human cells. There is a need to understand the human metagenomics. Therefore, to understand human metagenomics, we need to understand the bacteria population on humans. In 2007, NIH in America Microbiome project to coordinate our the research on human microbiome, and primarily the main conclusion that we obtained from these studies are that microbial diversity varies by an anatomic site. What does that mean? Microbiomes differ in terms of composition and function, and it, it is actually more site specific than across human population. What this means is the the microbial community, for example, in your mouth, is very different. It's probably more similar to the microbiome in another person's mouth, in contrast to other regions of your body. So there's more variety between your skin, the microbiome on your skin, and than in your oral microbiome, than when compared to other people. So microbiomes differ and is actually influenced by a person's health, genotype, diet, and hygiene. So the local environment and the biology of each body habitat is important. Then the main four phyla that is associated with human microbiome are the actinobacteria, firmicutes, proteobacteria, and Bacteroides. So some of the interesting results that we obtained, from from microbiome studies, show that when you take oral antibiotics, 1/3 of your gut microbiome is suppressed, and it only returns weeks after you stop the oral antibiotic treatment. However, several bacterial taxa remain undetectable. So, taking antibiotics is actually uh, very impactful on your microbiome. That's why nowadays, due to the increase in antibiotic resistance and due to our understanding of how microbiome is important for our health, uh, there is um, more hesitation on the doctor's part to give antibiotics. So if it's not a serious disease, probably the doctor will not prescribe any antibiotics. Then the alteration of a host environment, example via pH or nutrition, can result in changes in the microbiome. So either an increase in pathogenic bacteria or an upregulation of virulence genes. So this can lead to disease states. Prime examples are in your oral microbiome, oral cavity, where your dental caries are influenced by alterations in pH, and this is instead related to uh, your nutrition, the amount of sugar you consume, probably. Okay, bacterial vaginosis also can be related to pH and also related to amount of sugar, type of food that is consumed, and also related to stress levels of the host. So in the intestinal cavity, cavity, you have lots of microbes that are actually found interacting with your human cells. Okay, where the intestinal bacteria produce nutrients and molecules that actually modulate uh, mucosal immunity. So now our understanding has come to a stage where we know not only are the bacteria present in on our body, they actually uh, interact. Okay, there is some signal exchange between the microbes and humans. So when we are talking about oral microbiome, from the laboratory and cell culture studies, uh, they, the, their findings show that Porphyromonas gingivalis and Gregory T-Bacter actinomycete commitants are important for periodontal disease. However, when they looked at the population, they could not find any major difference in the bacterial species between the healthy and disease states. Okay, although they saw more bacteria associated with periodontal lesions. A possible reason for this lack of correlation could be that the population study, the sample size is too small. Okay, so need a larger sample size will be better for microbiome studies. And when they talk about microbiomes in the gastrointestinal tract, they find a connection between biomes and metabolism. Okay, what you eat, your diet, will shape the microbiome in your gastrointestinal tract, which in turn will affect the ability of the individual to absorb and metabolize nutrients. So from mouse model study, they found that the obese mice have more firmicutes, whereas lean mice have more bacteroides. Interestingly, when they transplant the gut microbiome from the lean mice into the obese mice actually normalized its body weight. Okay, so when you a possible solution to obesity could be actually to change the microbiome in order for the individual to be able to absorb and better metabolize the food that they take. So conclusion is microbial composition can affect body metabolism and energy harvest and influence predisposition to obesity. So this kind of animal study have actually been reported in human individuals. Other than metabolism, the microbiome in the gastrointestinal tract have microflora, example Helicobacter pylori, which is associated with gastric cancer and also uh, gastric ailments. Okay, so elimination of Helicobacter pylori was at first thought to be helpful because this would uh, eliminate peptic ulcer disease and also remove the possibility of gastric cancer. However, researchers have found that when you eliminate Helicobacter pylori, changes occur in the esophageal microbiome that actually increase risk of Barrett's esophagus and esophageal carcinoma. So by changing the microbiome here, by removing Helicobacter pylori, you may reduce risks of peptic ulcer disease and stomach cancer, but you increased other types of cancer. So it is not so straightforward as to just changing the microbiome. More studies are usually needed due to the wide variability in human population. Microbiome and gastrointestinal diseases, a popular example is the Clostridium difficile. Okay, this is a primary etiologic agent, a causative agent for the antimicrobial associated diarrhea. So this is a persistent diarrhea that occurs uh, in some patients that have undergone antimicrobial treatments because primarily because Clostridium difficile can overcome antimicrobial treatments, so it is very difficult to remove Clostridium difficile. So when the other microbiome is removed through antimicrobial treatment, this creates opportunity for Clostridium difficile to proliferate, and this causes disease. Okay, and it reoccurs. Okay, within days and weeks. So this is very difficult to remove, and to a state that doctors in America have now adopted fecal transplantation to restore the microbial balance and species richness in patients with recurrent Clostridium difficile disease. So usually when patients have recurrent Clostridium difficile disease and there are all the treatment options have been used up, fecal transplantation can be recommended. The feces is actually from a close relative due to diet concerns. So similarity in diet, they so that you can get a similar microbiome. So the feces from the close relative will be treated, cleaned uh, to obtain the microbial population, and then via colonoscopy, it will be transplanted onto the patient. And this is actually uh, a standard procedure in some countries because it actually works, and the patient be will feel much better after this treatment. Finally, vaginal and urethral microbiomes. So what when they studied the vaginal and urethral microbiomes, they found that vaginal microbiome has implications for pregnancy, preterm birth, sexually transmitted disease, vaginitis, and bacterial vaginitis. Okay, so the microbiome is important because it it is the changes in the microbiome here can influence the possibility of these diseases occurring: STD, vaginitis, and bacterial vaginitis. Vaginal microbiome also influence the neonatal oral and gut microbiomes through vertical transmission. This occurs during the process of giving birth, as the baby passes through the urethral and passes through the vaginal tract. It The baby is coated with the microbiome. For the for how the vaginal microbiome can affect vaginitis, it seems that when the microbiome has an absence or reduced lactobacilli population, this will predispose the vagina to a recurrent urinary tract infection. So why is the lactobacilli reduced or absent? Can be related to the diet or stress, and also which will in fact in turn affect the pH of the vagina.